Okay. Good morning, everybody. Thank you for coming so early and on the last day of the conference. First, I would like to thank the organizers for inviting me here to that fascinating conference, and especially that meeting, and to that wonderful town, Lyon. That is also the birthplace of Antoine de Saint-Exupéry, who is one of my favorite writers. And I'm going to speak about Petri nets at the molecular and cellular level. So we have been working on using Petri nets for modeling biochemical systems since about more than 20. And we started the modeling of metabolic systems and moved now to model more signal transduction pathways and gene regulatory pathways and hybrid systems. Why? Because a lot of qualitative data came up, qualitative and quantitative data came up, especially for signal transduction pathways, and the experiments for signal transduction pathways, especially in medical application, could be very difficult because of many reasons. So Petri nets. The first take-home message I want to give you is that Petri nets have nothing to do with Petri dishes. So Petri nets were defined by Carl Adam Petri in 1962 in his PhD thesis. And from that time on, Petri nets were mainly applied in computer science for modeling distributed systems and software dependency and so on and so forth, also for modeling business processes and financing systems. And in 1993, the first paper on a biochemical application occurs by Venkataraman Reddy. And since then, many papers have been published on applications for metabolic system signaling pathways using discrete formalism, stochastic formalism, and so on. So what are Petri nets? Petri nets are bipartite graphs. So, and I forgot to say, Carl Adam Petri developed the Petri net formalism in order to uniquely handle systems with concurrent processes. And what he did is really to distinguish between the active part and the passive part of the system. So we see here the so-called places drawn as circles that are the represent the passive part of the system. Then we have the rectangles representing transition, the active part, and we have directed edges connecting always vertices of different type. So you know now the structure of Petri nets, and I was thinking, when I was thinking about what to present here in the meeting, I want to give an example for a modeling of an application of Petri nets at the molecular level and at the cellular level. And we will consider Salmonella infection. And the work I'm presenting here was mainly done by Jennifer Hanig in her PhD thesis, supported by the postdocs Jörg Ackermann and Alireza Beji, who is also here in the auditorium, and had also a poster on the conference, but unfortunately yesterday. But you can ask him. He's sitting in the back. Okay. So let me give you some PetriNet definitions. We have the transitions for the active elements that can be processes, reactions. We have the places that are the passive elements representing biological species, compounds, and they define also pre or post conditions according to the arcs or edges they connect to the transition. And we have movable objects, the so-called tokens, and that could be molecules, proteins, and that are discrete objects and are indicated by black dots or by a number. And the distribution of these tokens over all places in the network define a system state and is also called marking. And then we have the edges defining relations. They could be weighted to quantify the pre and the post conditions. Let us consider here a simple reaction. So we have a substrate A and substrate B coming together to the product AB, and that is a corresponding PetriNet. And that transition is enabled and can take place because the pre-places contain at least as many tokens as indicated by the edge weight. If there's no number here on the edge weight, then they have the edge weight of 1, and at least as many tokens on place B. And now the transition is enabled and can take place, and the tokens will be removed at the same time from the pre-places and as they will produced on the post place. And that takes place according to a specific firing rule. The firing rule here is untimed, and that are the classical place transition networks called PT nets. And we will consider in the first part these PT networks. And but we can also easily introduce here a time stamp, so to get time discrete networks, stochastic networks, and even continuous networks. Here we have to use ordinary differential equations and concentrations, so that goes into the kinetic modeling. So that are the most important things of the PetriNet formalism. And if you are interested in more details, we edited a nice book on that topic. Okay, so coming to the modeling at the molecular level, the question is to what extent we can predict the system's behavior without knowing the kinetic parameters. And that is the main advantage of PetriNets, that you can generate statements on the system's behavior without the kinetic parameters. And that is a big advantage because for many biological systems, you have not a sufficient amount of the kinetic parameters available. So, and we will consider Salmonella, Enterica, Serovar, Typhimurium. I will speak only about Salmonella. They are looking here quite nice, but they are dangerous. They can cause gastrointestinal diseases and typhoid fever. And we have every year millions of infections and thousands of deaths. Moreover, Salmonella serves as a model organism of bacterial infections. So that work has been done with experimental collaborators, Ivan Dikic and Simone Fulda from our university. So we had the big advantage to have experimentalists we can ask and meet. So coming to the Salmonella infection, So you eat or drink something that contains salmonella, and salmonella infiltrates the epithelial cells. And here you have these epithelial cells. Here are our salmonella bacteria. And there are two main mechanisms known. Salmonella can form a salmonella-containing vacuole and replicate inside, but salmonella can go directly into the cytosol without forming such a vacuole. And you see here these red chains, and that are ubiquitin chains. They label salmonella for a further degradation. And that means that cytosolic salmonella gets ubiquitinated, and antibacterial autophagy started in the epithelial cells. And that is called xenophagy. Okay, so the aim was to develop a first computational model of that xenophagic capturing of salmonella. We wanted to verify the model by invariant analysis and in silico knockout analysis. And we also want to predict the dynamics of salmonella infection in the epithelial cells. So here I will just give a very brief overview on the pathways known for xenophagy of salmonella. So we have here four different ways, and the first one is a galactine-8 dependent recognition, and we see here that is the autophagosomal membrane, and if salmonella binds to that membrane, the degradation of salmonella starts. And here we have the damaged salmonella containing vacuole membrane. So the next pathway is galactine-8 dependent and ubiquitin dependent, forming big complexes that enables also the binding to the autophagosomal membrane. In the third, that pathway is only ubiquitin dependent. You see there are much more processes involved, but I will not go into detail here. And in order to bind to the autophagosomal membrane, and we see here that the damaged salmonella containing vacuole membrane is not part of that pathway. And the fourth pathway is a nutrient-dependent regulation of xenophagy, meaning that if salmonella enters the epithelial cell, we have an amino acid starvation that induces an amino acid normalization going through different complexes, also to ensure the binding of salmonella to the autophagosomal membrane. So that's all, and that is a corresponding Petri net. Just to give an orientation, here we have the pathway A, B, C, and D. Here we have the salmonella containing vacuole. And here we have the cell response, meaning the degradation of salmonella. And all is going through the binding to the autophagosomal membrane. And the places represent proteins or protein complexes. And we see here places of the same color and with the same name that are so-called logical places, meaning that these logical places of the same name are copies of one vertex in the underlying Petri net graph. So it's just for more clear drawing of the pathway. And that is the corresponding Petri net. Here we have the reactions defining the interface to the environment. And that model is also published in the biomodels database and also published in a paper. So what we can do now, I will speak now about analysis. So here I will not simulate, but I'm interested in getting a general information of the system's behavior, meaning which functional modules occur at every stage of the system. Then substance conservation, we will consider signal flows and in silico knockouts. So we define invariants at a steady state. First, place invariants for substance conservation, so that are multisets of places whose weighted sum remains constant, and that is an indication of substance conservation. And the transition invariants define functional modules and basic sub-networks if we consider the places and the edges in between. Transition invariants are multisets of transitions whose firing always reproduce an arbitrary initial marking. And they have been defined by Kurt Lautenbach very early, but had no importance or only a low importance for PetriNet theory. And 20 years later, Stefan Schuster could show that these transition invariants, he called them elementary modes, define basic functional modules in biological systems. And that was a great step in systems biology. And for the definition, for the mathematicians among you, so both invariants are based on the incidence matrix, and we are interested in minimal, non-negative, non-trivial integer solutions. Okay, just to exemplarily show you such a place invariant. So we have here a place invariant that describes the conservation of nuclear dot protein 52 and consists of 27 places. That makes sense in biology. So for that system, we generate, so we found 12 place invariants, all conserving a specific substance. So here we have exemplarily transition invariants. So the red one defines the ubiquitin-dependent xenophagic recognition for free cytosolic salmonella. The blue one, the regulation of the mammalian target of rapamycin 1 complex. And here the green one is a trivial transition invariant. And what you see here now, the red invariant really goes from the beginning, from the formation of the salmonella containing vacuole or for entering salmonella into the system until the cell response. But the blue one does not, and also the green one also does not. But we are interested in getting all signaling flows really from the receptor to the cell response. And that's why and that was an observation that transition invariants cannot provide that information. And that's why we defined manatee invariants. The name is caused just by the cute animal, nothing else. And the aim was to develop a mathematical concept to predict and characterize all complete signal flows. And here we have a small system in black, the first transition invariant, and that one is the first one going from the substrate to the formation of the enzyme substrate complex and then to the product. And here the second one is a synthesis of the enzyme and the degradation of the enzyme. And you see here that the firing of that transition invariant, so of the transitions in that transition invariance, cannot take place because we have no token here on the place for the enzyme. So we really need also that transition invariant. And what we did, I will not go here in very detail, but we just perform a linear combination of feasible transition invariants. That means that are reliable invariants in the initial marking, meaning that the transitions of that all transitions of the transition invariant can take place and can fire. And we have some conditions, so the property of feasibility is has to be conserved. The Petri net should still be covered by transition invariants, and importantly, we have the induced subnets of the Manatee invariant do not contain a place invariant anymore. Okay, and that is here now the one exemplary Manatee invariant we found for the system 19 Manatee invariants, and we see here nicely that we combine also the former blue transition invariant and former green transition invariant to describe ubiquity-independent xenophagic recognition independence on the regulation of mammalian target of rapamycin complex 1 and the cytosolic amino acid level. So, and that type of information, so place and transition invariants, we are using to verify the model, to really show somehow the completeness, but also the correctness of the model. We do that mainly manually. It's difficult to make the biological interpretation computationally. And another point is the in silico analysis that is also invariant based. And the basic idea is that all transitions invariants that contain the knockout transitions are affected by the knockout. And so here you see again a small system, and here you see the knockout matrix, where we compile color-coded the results. Here we have the transitions to be knocked out, and here we have the places. And that is a very easy example, yes. And you see if we knock out the synthesis A transition, then A cannot be produced anymore, and also AB not, but B can be produced. So, and that, we color coded it green for the unaffected places and red for the affected places. So, and that is a whole knockout matrix of our xenophagy Petri net. And maybe you can see that we have here some numbers in the elements of the knockout matrix written, and these numbers indicate the link to papers that experimentally proved that knockout. And that is a second type of validation. And what you can see here, you see that we have also generated many, many hypotheses which could be performed experimentally. So the knockout matrix gives you a compilation of the results of here it is done of a single knockout, but you can easily perform a double knockout or double knockout or triple knockout and so on and so forth. So that is the first part. I just want to mention for the project, so the modeling of the TNFR1 mediated signaling pathway has been done by Leonie Amstein in her PhD thesis. We considered the canonical and non-canonical NF-kappa-B pathway and their crosstalks. And we released 3 models to the biomodels database, and that has been done by Kira Trares in her master thesis. And at the moment, Markus Kessler, who is postdoc, is working on the IL-22 and IL-6 signaling pathway. He developed first a Petri net model, and now he develops a kinetic model on that. And that is all in close collaboration with experimentalists. We are in such consortium on acute, on chronic liver failure. And that is very promising. The results are still not published. And very recently, we published a review on PetriNet modeling of signal transduction networks for medical applications. Okay, so I'm coming to the stochastic part. So the question is, can we use stochastic PetriNets to predict the system's behavior at the cellular level? And for the stochastic PetriNet, the transitions are associated with stochastic rate constants and describe a random process. And the places is then, each place is a random variable, and the tokens on a place describe discrete values a random variable can attain. Here, in our example, the number of salmonella. And the simulation we are doing just with the well-known Gillespie's exact stochastic simulation algorithm. So we could also use other tools, of course, but first we did that classical PetriNet modeling to understand the processes, to verify the system, and to understand the biology behind. And then we started the stochastic PetriNet. So, and we want to consider the infection of an epithelial cell with salmonella, and we have here several processes: the landing, the takeoff. Here we have also a near surface swimming. Then we have a ruffle initiation, and also a ruffle joining to build a huge ruffle, and through that ruffle, salmonella goes into the epithelial cell. Here we have the cytosolic salmonella that could be degraded by xenophagy, and here we have the salmonella in the vacuole, and where salmonella replicates, and it depends on the number of salmonella whether the cell death is going on or not, and that is the washing is more technical process for the experiments. So, and that are the processes we are we will consider, and we build such a PetriNet, and we can see here a part of the PetriNet. So here we have the landing. Yeah, maybe you can read it a little bit. Here we have the initiation of ruffles. Here we have the ruffle joining. Here we have the proliferation of salmonella in the cytosol, and here we have the proliferation in the salmonella of the vacuole. And for that we need to find the parameters. And for salmonella, it was convenient, or we found many publications where we get the experimental data. We had to we needed for the stochastic modeling. And here I just want to show you the validation of our stochastic PetriNet. So here we have the simulation of the infection dynamics, and first 9 minutes infection time, and here 20 minutes infection time. So the simulations are drawn as a red curve, and in blue, the simulation is a blue curve and the experiment is a red curve. And here we consider the percentage of cells with membrane ruffles. Here's the percentage of infected cells, and here's the number of salmonella per ruffle, and here's the number of invaded salmonella per cell. And you see that it fits quite well. So we concluded our parameters are okay, and also the network is okay. And the experiment was the experimental data we took from Misselwitz and co-workers. So the same we did for the number of cytosolic bacteria per cell, and we have here also a nice agreement of experiment and simulation. So we have here the number of bacteria, 1 to 5, 6 to 10, and greater 10. And here we have the in percentage the cytosolic salmonella. Based on that, we predicted the intracellular bacterial composition. So first, I would like just to tell you about the heterogeneity of the cells. So each cell can only contain vacuolar salmonella or cytosolic salmonella or both. And we simulated the system 2 hours post-infection, at 8 hours post-infection, and 16 hours post-infection. And we see here after 2 hours, we have both the vacuolar and the cytosolic salmonella. After 8 hours, we have mostly the vacuolar salmonella. But if we have more than 100 salmonella per cell, we get also cytosolic salmonella. That is known. But what was not known is that after 16 hours post-infection, we get vacuolar salmonella that was not yet experimentally proven. So that is, as I understood, that is not so easy to prove it. And another prediction, what is interesting for the experimentally working scientists, is a prediction of the infection efficiency. So here we have the infection efficiency in percentage. Here we have the infection time. And here we have the MOI, so I forgot to introduce it. That is a multiplicity of infection, meaning the average number of salmonella in the infection medium that is added. And we see here for different MOI values in different infection time, which infection efficiency you will get. And that is important for experimental, for planning of experiments, and for example, how to really prepare a cell culture. So we have uploaded a paper under bioRxiv, and the paper is also under review in a journal. So that was the part for salmonella, and I'm going now to further projects at the cellular level. And here's a very nice project where we model the human lymph node. That is important because there are existing models on the mice lymph nodes and rat lymph node, but not the human one. And we are integrating own experimental data. So Sonja can read these paintings from developed by Martin Leo Hansmann, the pathologist in the group, and she translated that painting into a Petri net. Here the compartments are color-coded, so I will not go here into detail. And she also involved own experimental data, meaning imaging data of the human lymph nodes at specific disease states. That is a very interesting collaboration, and she did that with PetriNet. And a very recent work performed by Marius Kirchner, or he's still working on that. His idea is to combine PetriNet with agent-based modeling. So each bacteria is an agent, and each bacterium has an own metabolism. So, and each bacteria has an agent-specific behavior that is controlled by the metabolism that we get by a PetriNet simulation. And that controls that each specific behavior. That could be replication, movement, death, or other behavior. and that determines in the end the system's behavior of salmonella or bacteria containing systems. So, and we use the property of agent-based models that we have individual properties for each agent. And that is ongoing work and is not published. He's also involved in the acute on chronic liver failure consortium and also in the host pathogen consortium called ENABLE. Okay, so I'm coming to the summary. So first I introduce xenophagy capturing salmonella. So that are the molecular biological processes, and that is the corresponding classical place transition network. And then I introduce the cellular biological model for salmonella infection, and we performed a stochastic salmonella Petri net, which we simulated, and here we could show a good agreement with the experimental data. And here we here we verified the model with using an invariant analysis and also the in silico knockout analysis. So take-home messages. So what we learned in all the years, please always keep in mind that the quality and the quantity of the data determine the modeling method. So that means you have to check your data very, very carefully. That was also mentioned in other talks here at the conference, also in the keynotes. And another point is, even if you are a computer scientist, you have not only to understand the biology, that is the first point I want to make, but you have also to understand the underlying experiments. Go to your collaborators, ask them to show you the experiments. Maybe you can perform also by yourself one small experiment. And another point, I wanted to convince you that the system's behavior can be predicted without knowing kinetic parameters to some extent. That is clear. For example, using Petri nets, but you can also use Boolean models or logic models. But for Petri nets, the invariant analysis is very useful, knockout analysis, and Petri nets are semi-quantitative, so you could also simulate the Petri net. I did not show it here, and what is quite interesting in that context is the reachability analysis. It's still not possible to really get all possible systems of the states of a system. So then, of course, you have to validate your model. You can do that computationally, but the experiment proves your model. And discuss your results as often as you can with the wet lab scientists. That is so important. And I learned from Burkhard Rost, do not forget that science is communication, and I want to extend it to science is face-to-face communication. Okay, I'm coming to the acknowledgements. So I'm really grateful to Stefan Schuster and Falk Schreiber. Stefan Schuster was the one who gave me, I think 25 years ago, the ready paper on Petri net applications for modeling metabolic networks. And we had a lot of discussions with him and also with Falk Schreiber, and that our experimentalists at the Goethe University, Ivan Dikic, Simone Fulda, Maria Wereschild, Christoph Welsch, and Heiko Mühl. Both Ivan Dikic is a specialist for ubiquitination. Simone Fulda unfortunately left the university and or science. She is now the president of the University of Kiel. Maria Wereschild is a physician for infection biology, and Christoph Welsch and Heiko Mühl are working on the interleukin pathways. And I'm also very, very thankful to Denis Tier-Free and Anania Rakes. We had quite nice discussions. And of course, I thank all the institutions here for financing and supporting the project. But my biggest thank is going to my fascinating group. And you see here maybe Jörg Ackermann, Markus Kessler, here's Ali Reza, who is sitting in the back. Florian Giston is also here. He's working on modularization of protein complexes using graph theoretic method. He had also a poster here at the conference. Here is Sonja again, and it is always very enjoying, very stimulating to work with the students and co-workers of the group. And for you, I'm saying merci beaucoup for listening me, and I'm happy to take your questions.
We have time for a couple of quick questions.
Oh.
Thanks for the great talk. I have a question on the Petri nets in general. So you said, of course, the data quality and quantity decides which method you use, but are there also special modeling aims where Petri nets are very well suited to?
Modeling aims, so first to verify the model, yeah, then also to analyze the model, not just to simulate the model from a specific initial state, meaning an evolution of the model, but to analyze the overall behavior that could take place in principle, yeah, and that is, I think that was your question, so your main question, and the point is, you can of course you have a lot of data, but you have much, and in most cases you have much more qualitative data. And if you want to find the parameters for the quantitative modeling, so for Salmonella, we had the luck to find it in the literature. But otherwise, you have often to perform a parameter optimization, what is quite difficult. You are an expert, I think, in the group. And before starting that, and before starting an ordinary differential equation modeling, you can make sure that your model is complete and somehow correct.
Yeah, very cool. Thanks.
Yeah, so I have a follow-up question. Thanks from my side also. You showed one example where you had the stochastic Petri net, and you also said that there you did the parameterization, I guess via optimization. And so my question is how you did that, whether this was difficult, and which kind of data you used. I think it's fold change data, right? Rather than concentration.
Yeah, that are fold change data. But we used the data just from the experiments. But if you want to have detailed knowledge, Ali Reza is sitting in the back, and we can discuss it later. But it's all from all published, experimentally proven data.
Okay, thanks.
Thank you for the very interesting talk. So I was wondering, so the Petri nets give some kind of validation on the model completeness, right?
Yeah.
And you presented two models of Salmonella from the molecular and cellular level. Have you obtained some insight from the Petri net modeling of the molecular level that you used then for the creation of cellular level and the stochastic model?
That were different processes here.
So
we modeled. That was not directly related. But what was the first part of your question?
It was basically that I was wondering whether.
But what was new, so we detected really new complete pathways, and the in silico knockout analysis gave a lot of hypotheses. So we have contacted also groups, experimentally working groups, but that will take a lot of time. Sure, but yeah.
Thank you very much.
There's
Very quick. And then we can use the computer.
Just a quick question. So the Petri said you have a time dimension, right? So you iterate.
For the analysis in the classical place transition networks, we do not consider time. So tokens will be removed from the pre-places in the same at the same moment as they will produce on the post-places. But for the simulation, of course, there is a time component.
And sorry, a quick follow-up. And which aspect of the ODE system would they then contain? I presume it's just the steady states, right? So they'll ignore stuff like limit cycles and so on.
For the kinetic modeling, so I'm using ODEs, and you can integrate the ODEs into the firing rule. But I would always advise to use Copasi or other modeling tools for ODE system simulation, because that is much more efficient, much better. Yeah, but in principle, that is possible. I just wanted to show that it is very easy to introduce new concepts because of the consequent partitioning into an active part and a passive part of the system. The main idea of Carl Adam Petri.
Thank you, Lina. You can